Welcome to this lecture on biological databases, the absolute foundation of modern computational biology. Today, we are going to explore how scientists store, manage, and search through the massive amounts of genetic data being generated every single day. Whether you are looking at DNA sequences, protein structures, or complex metabolic pathways, understanding these databases is your first crucial step into the world of bioinformatics. By the end of this session, you will not only understand what these databases are, but also the mathematics that govern their explosive growth and the statistics we use to search them effectively. To begin our journey, imagine you are trying to find a specific chocolate chip cookie recipe in a physical library that contains 1 billion unorganized books. You would spend your entire life searching and likely never find the exact page you need. This is exactly the problem that biological databases solve for modern scientists. We have sequenced millions of DNA strands and proteins over the last few decades. Without a structured, digitized way to store and search them, this massive mountain of data would be completely useless. Biological databases act as that lightning-fast search engine for the building blocks of life. Now that we understand the problem, let us define what we are actually working with. A biological database is a highly structured, searchable digital archive. It is not just a simple text file. It is a complex system designed to store intricate biological data, such as DNA sequences, three-dimensional protein structures, and entire metabolic pathways. These databases allow researchers from all over the globe to access the exact same standardized information instantly. They transform raw biological molecules into readable digital code that computers can analyze, compare, and manipulate to help us understand the fundamental mechanics of living organisms. Building on that definition, we must recognize that not all databases serve the same purpose. These databases are generally divided into 3 main categories based on how the data is processed and verified. First, we have primary databases, like GenBank, which act as raw archives where scientists directly submit their newly discovered sequences without much alteration. Next, we have secondary databases, like SwissProt. These take the raw data and add layers of expert analysis, curation, and biological context to make it much more meaningful. Finally, composite databases act as massive search portals that seamlessly pull information from multiple primary and secondary databases at once, giving researchers a unified view. Understanding these categories leads us to why this infrastructure is so critical. The importance of biological databases cannot be overstated, as they form the absolute foundation of modern computational biology. Consider what happens whenever a new virus emerges. Scientists immediately sequence its genome and upload it to a primary database to share with the entire world. Researchers can then compare this new virus against millions of known sequences to find out where it came from and how to build a vaccine. Without these centralized databases, global scientific collaboration would be impossible, and life-saving drug discovery would grind to an absolute halt. With this global collaboration in mind, we must address a massive logistical challenge. To truly understand the scale of biological databases, we must look at how rapidly they are expanding every single day. Because DNA sequencing technology has become incredibly cheap and fast, the amount of data submitted to databases like GenBank does not just grow steadily, it grows exponentially. We can model this massive explosion of biological data using standard mathematical functions. This mathematical modeling is crucial because it allows computer scientists to predict exactly how much server storage and processing power will be needed in the future to keep the databases running smoothly. To quantify this rapid expansion, let us look at the mathematics behind it. This is the standard exponential growth equation used to model database size. Conceptually, it shows how a quantity grows continuously over time based on its current size. In this formula, n of t represents the total number of sequences in the database at a given future time. N0 stands for the initial number of sequences present right now. The constant e is Euler's number, approximately 2.718, serving as the base of natural logarithms. The variable r represents the continuous growth rate, and t represents the elapsed time in years. If the growth rate r increases even slightly, the future database size will explode much faster, requiring massive and immediate infrastructure upgrades. Now that we have our formula, let us see this with a concrete example. Suppose a primary biological database currently holds exactly 100 million DNA sequences, and based on historical trends, it is growing at a continuous rate of 20% per year. We need to calculate how many sequences it will contain after 5 years. First, we identify our known variables and plug them directly into the equation. We substitute 100 million for n0, 0.20 for the rate r, and 5 for the time t. Multiplying 0.20 by 5 gives us exactly 1 in the exponent. Since e to the power of 1 is approximately 2.718, we multiply our initial size by this constant. The final answer tells us the database will contain approximately 271.8 million sequences. Physically, this means the bioinformatics team must prepare their servers to handle nearly 3 times the current data volume in just half a decade. Having calculated the future size, let us visualize what this growth actually looks like. Here, you are seeing the exponential database growth curve. The horizontal axis represents time in years, while the vertical axis represents the number of sequences in millions. Notice how the blue curve starts off relatively flat, but then suddenly sweeps upwards in a steep, nonlinear fashion. This curve perfectly illustrates the mathematical reality of our previous calculation. As time moves forward, the rate at which new sequences are added accelerates dramatically. This visual teaches us that managing biological databases is not a static task. It is a massive, ongoing computational challenge that requires constant hardware upgrades just to keep pace with the science. This massive scale we just visualized introduces a completely new challenge when we actually try to use the data. Because these databases are so massive, searching through them creates a mathematical problem regarding statistical significance. If you search a database containing billions of sequences, you are bound to find some matches purely by random chance, just like winning the lottery. A random match does not mean the sequences share an evolutionary history. It is just background noise. To separate true biological relationships from this random background noise, scientists cannot just rely on raw alignment scores. They need a rigorous statistical metric to evaluate how trustworthy a search result actually is. To solve this problem of random noise, scientists use a metric called the expectation value, or e-value. Conceptually, the e-value calculates the exact number of random matches you should expect to see when searching a database of a specific size. In this formula, e represents that expectation value. The variable m represents the length of your query sequence, while n represents the total size of the biological database. The variable S is the raw alignment score, which measures how well your sequence matches the database sequence. Finally, K and lambda are statistical constants that depend on the specific scoring system used by the search algorithm. Notice that if the database size n increases, the e-value increases, meaning you are much more likely to get random, meaningless matches in a larger database. Let us apply this formula to determine the statistical significance of a real database match. Suppose a researcher searches a 100 amino acid sequence against a database of 1 billion amino acids, getting a raw score of 50. The constants K and lambda are 0.1 and 0.5. We want to know if this match is biologically meaningful. First, we substitute all our known parameters into the e-value equation. We simplify the search space portion by multiplying 0.1 by 100 and then by 1 billion, resulting in 10 billion, or 10 to the power of 10. In the exponent, multiplying negative 0.5 by 50 gives us negative 25. The term e to the negative 25 represents the tiny probability of getting a score of 50 by pure chance. Multiplying our massive search space by this tiny probability gives us a final e-value of approximately 0.138. Physically, this means we expect nearly zero random matches of this quality. Now that we have calculated the e-value, let us understand what it actually tells us. The final calculated e-value was approximately 0.138. This means that in a database of this massive size, we would only expect to see 0.138 matches of this quality by pure random chance. Because this number is much less than 1, the researcher can confidently conclude that the match is biologically significant. It is not a random coincidence. The two sequences likely share a true evolutionary history. This proves exactly how vital mathematics and statistics are for interpreting biological database searches. Without these calculations, we would be lost in a sea of false positives. To wrap up our lecture, let us review the most important ideas we have covered today. First, biological databases are not just static filing cabinets. They are dynamic, rapidly expanding ecosystems of information that form the backbone of global scientific collaboration. Second, we saw how exponential growth models are essential for predicting the future computational infrastructure needed to maintain these databases. Finally, we learned that searching these massive databases requires statistical tools like the e-value to separate true biological relationships from random background noise. Understanding how these databases grow helps us build better computers, while understanding the statistics of searching helps us find cures for diseases. You have now taken your first and most crucial step in becoming a proficient computational biologist.